Okay, I have it at 4:04, so how about we get started? I want to welcome everybody to the SPARC FAIR Codeathon Showcase. This is a celebration of the projects that were done this August, and it represents an opportunity for the SPARC program to encourage and invite development of novel technologies and interactions with the data that are stored on the SPARC portal in a short 3-day codeathon. And so now I'm going to hand it over to Hiba Benaribi, who will present their team's project, Transcriptomic OSPARC. We'll hold questions and comments and discussion till the end, but we're going to start off with this presentation, and I will stop sharing to allow that to happen.
Hello, everyone. Hello, everyone.
Hi, Hiba.
I will share my screen. Do you see my screen?
We do.
Okay, so hello, I am Hiba Benaribi. Today I will be presenting on the behalf of T5, namely Transcriptomic OSPARC. So it's composed with me and Mod Kiran and Menjinding. So during this codeathon, we developed a template on the SPARC platform. We named it Expression with the goal to visualize the genetic expression data. So when you access our template, you will find 2 pipelines that were coded in Jupyter notebooks, and you will find 2 spaces named dataset 1 and dataset 2 for you to import your data. I will start with explaining the first pipeline. So as it was showing, it takes as input a CSV file for expression data. So the pipeline will first identify the differentially expressed genes based on the p-value and the log FC. As default, we identify a gene as differentially expressed if the p-value is less than 0.05, but it could be customized by the user to 0.01. And then we identify the genes as upregulated if the log FC value is positive and downregulated if the log FC value is negative. It also could be customized to less or more than 2. It depends on the user's preference. And then the expression profile will be represented in a volcano plot. So the non-differential genes will be colored in gray, the upregulated genes in green, and the downregulated genes in red. And the plot could be downloaded as a PNG file. Then the pipeline will perform the ontology analysis. So there's 3 ontologies. We do have the biological processes, the cellular component, and the molecular function. And you will get 6 plots as a result, including the top 20 ontologies. So we'll get in total 6 plots. You do have the biological processes for the downregulated genes, the cellular component of the downregulated genes, and the molecular function of also the downregulated genes. You will have the same plots for the upregulated genes, and also the plots could be downloaded as PNG files. So we do have the second pipeline. It takes as input 2 CSV files of gene expression. So it also performs the identification of the differentially expressed genes as the first pipeline. But in addition, it also compares the 2 data sets. So the first output, you will get a table that will identify the number of genes that do have similar expression profiles in the 2 data sets, and those that have different expression profile. And you could also export the result in a CSV files that will identify for each gene the expression profile in the first data set and in the second data set. It could be used if you do have 2 cells or 2 stages of a specific disease you want to compare. So how to use it? So for that, we provide a web browser extension that will guide you from opening the template to downloading your data. So initially, we developed this template to be associated with the SPARC Portal transcriptomic data. That's why the extension also guides you from the SPARC Portal, from finding transcriptomic data to downloading data. And in case no processed data was available, we suggest a pipeline that could be used to process the data. And it also provides a quick access to the template from a beta. The extension is available in the project GitHub repository. The folder could be downloaded and it could be installed using the mod developer. Yeah, the template is now public in the SPARC platform. Feel free to use it if it is useful for you. Thank you for your time.
Thank you, Hiba. Tell me, was this your first codeathon?
It was actually my second codeathon, but it's my first quick, short codeathon. So the first codeathon I participated in, I had actually a month to prepare for my idea, but this one was quick. We directly find an idea and start working on it. So it's a very different experience for me.
Oh, that's spectacular. Thank you for your presentation. Your team worked really hard. You created something that is really quite fabulous, and I'm really glad that it is available for others to build upon as well. Let's move on to our next group. Oops. And that will be Quilted Tutorials, which is an online tutorial hub to guide users within and outside the SPARC community on how to combine tools within the SPARC ecosystem to create unified workflows. This project will be presented by Yuda Monarco. Yuda, are you here?
Yes, yeah. Thank you, Susan.
Sure. I
Can you-
think you can share your screen and have at it.
Yeah. Okay. Hi everyone. Thank you, Susan and SPARC team for organizing this event. And I'm Yuda Monarco from Quilted Tutorial teams. And we have Omkar Atafali, Kenneth Tran, Matthias Russell, and Nilufar Shahidi. And the NIH SPARC initiative has developed an ecosystem of open source tools and carefully documented sets of data underpinned by the principle of FAIR, which are now prime for utilization by people within and outside the SPARC community. However, we see an opportunity to lower the barrier for entry into the SPARC ecosystem because many existing tutorials show in details how a particular SPARC tool can be used on its own, but do not document how these tools can be used together for building workflows. We have developed a hub for online tutorial with the goal of realizing the potential of the tools that are currently available within the SPARC ecosystem. We have populated the hub with tutorials that demonstrate how SPARC tools can be used for unified workflows. And then these initial tutorials lay the foundation for what we envision to be a series of tutorials hosted on our hub that serve as a place for new and current members of the SPARC community to learn about ways in which SPARC tools can be used collaboratively. We expect this to empower a broader user base to access and use SPARC data and tools and grow the SPARC community. Our platform will be a place for the SPARC community to actively contribute tutorials and share with others in the community. And our online tutorial hub is accessible via a website and provides a list of all available tutorials. Here we present a unified workflow that processes and visualize FAIR data from the SPARC data portal using open source tools. The workflow consists of data acquisition and data processing and visualization using open source tools. The tutorials are delivered through a Jupyter Notebook environment, which allows simple interactive code and user feedback in a familiar user interface. Our tutorials cover how to process and visualize this data using the open source tools. Our first tutorial consists of 3 steps: acquisition of 2D spatial distributed data from the SPARC portal, projection of the 2D data onto a 3D organ scaffold, and visualization of the scaffold. For data acquisition, we use the Pensive web API to download the required derivative data file, which describes the spatial distribution of three different types of neurons on the surface of the rat stomach. The next step is to project the 2D spatial information onto a 3D scaffold of the stomach, and then we use SPARC scaffold mapping tools to produce a WebGL encoded mesh of the rat stomach, and then which was then converted into STL and visualized within a Jupyter Notebook. The result of our tutorials is an interactive figure in Jupyter Notebook showing the projection of the 3 types of neurons on the surface of the rat stomach. Given the modularity of our generalized workflow, we can easily substitute alternative methods. For example, an alternative but more involved implementation would be to directly call scaffold mapping tools from within Python and make use of the rich built-in functionality for 2D data projection and 3D scaffold visualization. Then, in the second tutorials, we make use of the same general workflow, but instead of projecting 2D data onto a 3D scaffold, we demonstrate how the 2D data can be processed for computational simulations. In our second tutorial, statistical processes are used to compute a probability density estimation of neurons on the surface of the stomach. And then this density function can be used to simulate network of neurons that are generated using data-driven probability functions. In summary, we have produced a generalized unified workflow for acquiring experimental data and processing it using only resources from SPARC. And then we have demonstrated the feasibility of using just SPARC resources to drive this workflow using a rat stomach data as an example. And then a central benefit of this approach is that it leverages the FAIR principles that underlie these SPARC resources to ensure that the integrated models that are produced are also fair. And then our approach is creative because it encapsulates the workflow within a Jupyter Notebook online platform, and then which is designed to be readily accessible to new users of SPARC. Having a tutorial that can be easily engaged will not only accelerate growth of the SPARC community, but also increase uptake and usage of tools by members within the community. Thank you very much for having me.
Thank you, Yuda and Quilted Tutorials team. I particularly liked how you recognized how powerful the ability to teach by example or learn by example by creating some tutorials is a great way to provide a hopping off point for others to start to get their feet wet in how to effectively utilize the data that's present on the SPARC portal, as well as the tools that the SPARC Data and Resource Center have created, and integrate them together to generate novel analyses. So I really appreciate your well-crafted tutorials. Thank you. So next, we are going to see from SPARClet. SPARClet is a codeathon project that uses a Python library to generate and interact with flat maps in a Jupyter Notebook using the IPyLeaf or IPyLeaf library. Arkit, you are going to be the presenter for this project, I believe.
Yep, exactly.
Hand it over to you.
Yeah. So I think I kind of tweaked the presentation before, but it's fine. So thanks, Sue, for the introduction, and the presentations till now has been amazing. I was just wanted to say that before. So here I'm going to present to you the Sparklet library that we wrote, package Python library that we wrote. That was done by me, Arshit Bhatnagar, Chloe, and Kaustubh Sudarshan from all together in Team 4 on the SPARC Codeathon 2022. So what exactly is Sparklet? So as like Sue did half of my job here of introducing flat maps. So as Sue already introduced flat maps, these are these kind of maps that have been generated by SPARC to integrate the information for the peripheral nervous system from all the different data sets available. And if you go to the SPARC website, there's a nice interactive window that allows you to interact a lot with these maps. However, this interaction is limited in the sense that that's all you get. There's nothing more. However, there is in Jupyter Notebooks, there's actually ways to get geographical maps using IP leaflet and the corresponding Python package IPy leaflet into these Jupyter Notebooks, and then add interactions on top of these. The whole point of Sparklet, what exactly Sparklet is trying to do, is to take these SPARC flat maps and treat them as these geographical maps, put them in place of these geographical maps, and then allow customizable interactivity to be added in. So instead of just having the interaction that the SPARC websites allows you, you can actually add your own interactions, whatever you might think of. And so to do this, essentially what Sparklet does is it basically is constructed into two classes. One is a build map, which is the one that's taking the flat maps from, which is talking to the SPARC flat map servers, taking the flat maps from there and importing it and drawing it in Jupyter Notebooks. And the other one is the IPy leaflet add-ons class, which is containing all the interactive widgets that are already available. And this is the class that you can extend on to actually add new stuff. I'll first start with the build map stuff, and then show you later on what the leaflet add-ons can do. So starting with the build map, what essentially it does is that it sends a request to the flat map server, the server that's containing the different SPARC flat maps that are available. And that the server gives back us a list of map IDs saying, okay, these are the IDs for the flat maps that you want to access. Using that, then you can keep asking the server for different things, such as the metadata for the particular flat map itself, the anatomical annotations that Sue was talking about in her initial presentation, etc. The main thing that this build map thing requests is this two particular things. One is the tiles for the image layer, and one is the tiles for the vector layer. These essentially are the two building blocks of the flat map itself. And what this essentially allows you to do is within 4 lines, just by importing build maps from Sparklet, and then just building giving it the URL for the server and a particular tag identifying which map you want to use, you can essentially get the flat map immediately within 4 lines in your Jupyter Notebook. The function also provides, so for example, if you don't know this tag, the function actually provides you a way of telling that, okay, these are the flat maps available in the server, then you can select your tag based on that. That's what build map does. The cool stuff is done by the leaflet add-on things. What it does here is that it allows us to add all the interactive widgets. And some of these examples are here. So here is like the fully loaded version of the Sparklet and drawn flat map with all the interactive widgets. This is, I believe, for a cat. And what it allows you to do is to add custom drawings. So you can draw on this flat map in your own, like you can use these tools here to draw on the flat map to highlight certain regions. There are these annotated markers. These numbers that you see are essentially groups of annotated markers that are annotated based on anatomical information that we got back from the flat map itself. There's layer control, which allows you to control which layers of the flat map you want to see or not. There's a search bar, which allows you to then, so you can type in saying, I want to look for this particular anatomical feature, and it can allow you to search for that, and also additionally allow you to search for that particular analytical feature in other data sets, such as PubMed. There are custom markers, which act similar to custom drawings, and you can add them to highlight certain regions. And finally, hover facility, in the sense that you can hover over the map at a certain point, and it will tell you what anatomical feature you are looking at. So for example, if you hover over the brain, it says the brain. This, however, is not the limit of this particular system. You can actually do more. For example, one of the things that we were trying to build, which we could not build due to lack of time, was to allow you to compare two flat maps side by side. So you can have a flat map from, say, a particular flat map constructed from a set of data sets for rat, and another one constructed from another set of data sets for rat, or maybe for a cat instead. And you can compare them side by side. You can add heat maps. This is a feature that has already been implemented a bit, but still needs some information in there. So this can allow you to highlight certain features inside the map. And you can also add animated pathways, which can allow you to highlight certain tracks. This particular package is available in GitHub. And along with that, you also have some tutorials which guide you through this process. There are a bunch of Jupyter Notebook tutorials which guide you through the process of how to build a map, how to add leaflet add-ons. And finally, this project is also available as a Python library, so you can download it right now and start using it. And that's all. So thank you for your attention, and I would welcome any questions.
Thank you, Archit. That was spectacular. I really liked how you guys managed to pack in so many features in 3 days of effort, and still had time, I'm assuming, to eat and maybe get a couple of hours of sleep. You had a tutorial, you had well-documented code, and you brought in a third-party set of tools to augment the tools that were created by the SPARC DRC and utilize data that was present on the portal. It was very well done. It's very exciting to see.
Thank you all for, it's not just me, the whole team.
Yes, exactly right. You guys crushed it. The whole team did a great job. So we have one more presentation to go. And this is a presentation the project is entitled SPARC Me. It's a Python tool to explore, enhance, and expand SPARC datasets and their descriptions in accordance with FAIR principles. Prasad is going to be our presenter for this particular project. Prasad, are you ready?
Yes. Can you hear me, sir?
Sure can.
Awesome. So thank you very much for giving us the opportunity to, our team, the opportunity to present. I'll just going to quickly share screen and see if I can get this thing playing. Okay, so give me 1 second. There we go. Share sound. And hopefully you'll hear this. Hi, everyone, and welcome to the SPARC Me presentation. Sorry, just a quick thing. Were you able to hear? Okay, awesome. I'll press play again. Hi, everyone, and welcome to the SPARC Me presentation. Well-curated datasets are the foundation of the SPARC project. A SPARC dataset comprises of an experimental protocol and data files organized in the SPARC dataset structure, or SDS. This structure has been adapted from the brain imaging data structure, BIDS. The SPARC community have developed a step-by-step interactive tool, SODA, for creating datasets for curation. However, one of the problems is that there's no publicly available approach for programmatically accessing metadata fields in these SPARC datasets. And also, there's no publicly available approach for creating new SPARC datasets. So this limits the application of FAIR principles for researchers to interact with SPARC datasets. It also limits the development of novel tools that make use of these data sets. So our solution to this problem has been to develop SPARC.me, the SPARC metadata editor. And this is a Python tool that allows us to explore, enhance, and expand SPARC datasets in accordance with FAIR principles. We've created a number of guided tutorials that will showcase how users can use SPARC.me to help them in their research. This includes the ability to download existing SPARC datasets, create new SPARC datasets, interact with these datasets on OSPARC, and also how we can extend the SPARC dataset specification with descriptions, for example, from the data use ontology developed by the GA4GH. And this provides, this tutorial specifically provides an initial attempt on how we can start to use the SPARC dataset structure to store clinical data. We've also provided a tutorial as a first step towards how we can start to convert BIDS datasets to the SPARC dataset structure. In terms of impact, SPARC.me will help support the SPARC DRC and community developments. It will also help support interoperability with other initiatives. For example, we can try and use SPARC.me to help map the SPARC dataset structure to data dictionaries that are used in popular research platforms, such as the Gen3 Data Commons, which is used across a number of different research initiatives. We can also use SPARC.me to help enable extensions to the SPARC dataset specification to look at how we can start storing results from digital twins, which are being developed for precision medicine. I'd really like to thank our team for a great collaborative effort over the past few days. And I'd also like to acknowledge the SPARC program and also our SPARC Codeathon mentors. Thank you very much. Thanks, thanks everyone.
Thanks, Prasad. Before we move to an open discussion, I would just like to share a bit more information about what you guys were all working towards. There is, of course, the excitement of participating in a novel set of contests to see what you can do in a short amount of time with people you may or may not know, along with the challenge of working with data and tools that you have perhaps limited familiarity with. So there's always that excitement. But in this case, the SPARC Codeathon, with support from the National Institutes of Health, the SPARC Data and Resource Center was able to offer some prizes, and judging was administered by a number of experts. So we were quite excited to be able to offer an additional incentive for going the extra mile to create some great projects, which you guys all did. It was fantastic. So each project is judged on the quality and the completeness of the code, documentation, because if you can't explain it to me, how am I going to be able to put it into practice on my own? So we really put a lot of emphasis on how well you documented your code and documented the principles. And then, of course, the recorded presentation that summarizes your project for the judging panel. So each team presented a 4-minute recorded session, like Prasad presented this time, to the judges, and that's what the judges utilized for the judging of the individual projects. The criteria were the creativity of the team's solution, the potential impact on the SPARC research community, the feasibility based on prototype implementation, and as I said, that completeness of documentation. So the feasibility based on prototype implementation is something I just want to highlight, because all of these projects, I feel, presented really tractable and practical opportunities to use SPARC resources, the data, the resources that the SPARC DRC created, in a way that really belied the fact that you only had 3 days to generate this. These projects really look quite smooth to be perfectly honest. So kudos to every team member and every team. The prizes that were offered broke out into 4 cash prizes. And importantly, every team that participated in the Codeathon had the opportunity to submit their project as a manuscript, which we think is a great opportunity to help introduce, explore, and expound on techniques for interacting with FAIR data, as well as the opportunities that are afforded by working with the SPARC program. And so I'd like to take a moment to congratulate all teams and to run through the individual prizes. So the 4th prize team, or the team that received the 4th prize was the Transcriptomic OSPARC. Hiba presented that project first. She was our first presenter. Our 3rd prize, which amounted to $7,000 US to be distributed across the team, was awarded to Quilted Tutorials. And 2nd place went to SPARClet. So the leaflet project, and $10,000 was available to the team from SPARClet. And the grand prize winner is SPARC Me, the last presentation that we heard from. So Prasad's team all together have the opportunity to distribute amongst themselves $20,000 US. So we really appreciate everybody's effort, excitement, dedication, and the fact that that you were really committed to creating something really cool. I know that a number of projects or a number of teams were working on manuscripts and tidying up their code even after the Codeathon, which I think is a really unusual, perhaps, aspect of this particular Codeathon. And so I'm really excited and hopeful to see the implementation of the final project. All, as was mentioned, all of these projects are available on GitHub. They are located on the SPARC portal, so that you can navigate to SPARC.science and access from there the videos, the tutorials, as well as the GitHub repositories, so that you, as you're interested, can dive into those individual projects and extend them. We will offer another Codeathon in the future, and there is no reason you couldn't continue to expand on these projects. So I'd like to open it up for discussion. And just to get things kicked off, I want to circle back to Prasad really quickly, because he spent a little bit of time telling me about some of the things that they were working on to continue developing their Codeathon project, SPARC Me. So Prasad, do you want to tell us what you've been up to? You and your team, of course, not just you.
Yep, for sure. So I can maybe just give a quick overview of why, one of the motivations for SPARC Me, and also how we're trying to continue developing it. So I'll try not to spend too much time here, but maybe I'll just share my screen and just show that last slide here. So, so some of the things we're hoping to do with, at Auckland Bioengineering Institute, we have this project called the 12 Labors Project. And what it's aiming to do is, one aspect of that is to create a platform for precision medicine. And the idea here is that we want to be able to pair clinical measurements from an individual, or just measurements from one specific individual, and pair that with models of different organ systems. And what we're trying to do there is develop these automated workflows that will effectively allow you to personalize the models with the data. And so these different workflows are being developed to address different, help with diagnosis and treatment of different diseases. But as part of this, what they're actually doing is they're creating these personalized models, and sure, they're creating them for different, different parts of the body. And what we ultimately want to do is take all the outputs from all of these workflows and integrate them together to create digital twins. So that's what this project is doing. And what SPARC.me is really trying to help, will really help us do is figure out how we can store data from these workflows. And we really want to use the SPARC data set structure. It's very well designed and it's very well designed and actually, satisfies FAIR principles. So we want to be able to make use of that to store the outputs of these workflows. Now, we want to make these workflows automated. And this is why this was one of the main motivations to try and create SPARC.me in a way that allows you to programmatically create data sets. As we run through these workflows, maybe we can create data sets and use these SPARC dataset structures to help with trying to piece together the digital twin puzzle. We're also hoping to work with the SPARC community to see how we could extend these SPARC dataset structures to store sensitive human data. The other aspect was also this supporting interoperability. So we really want to make use of the data in the SPARC the SPARC data to create these digital twins. And we also want to be able to access data from other initiatives, and maybe SPARC.me might be a tool that might be able to help link these communities. Thanks, Sue.
Yeah, thanks, Prasad. I think that's really cool that you saw an opportunity in this this short codeathon project to incorporate it into your own extended research activities. I think that illustrates the benefits of going FAIR, right? Because if you build the tools to be findable, accessible, interoperable, and importantly, reusable, then hopefully they themselves have that opportunity to be extended and derived into other forms that allow other projects to make use of that. So I think that that's really, really cool. So while we have a bunch of people on the line, would anybody like to ask any questions of the teams? There's the presenters and a number of the members of each individual team that are on the line here. So if anybody has a question, please. David Nickerson has a question. He's raised his hand.
No one knows David. I mean, just to firstly emphasize everything Sue said, that we're just blown away by how much everyone was able to achieve in 3 days. It's ridiculous. But I guess my question is, I mean, I know the DRC side asked sort of thing, and I can kind of get an idea of how much work everything was to do that. But the one I have no idea about was the Chrome plugin. And I think that was a great idea on a way to try and help guide and provide this sort of extra information. So I think Heba, sorry. I just wanted to ask, was the Chrome extension that you guys came up with, is that something you or someone in your team had any experience with before the Codeathon, or that was also like a completely new technology to work with?
So hi. No, I actually had no idea before this Codeathon how to develop an extension, but just we wanted to provide something like a tutorial. So I thought about an extension in the time and learn it at the same time how to do it and develop it.
That's
crazy.
Really exciting. I can't believe that. So I want to ask, I think that that's really cool that the Codeathon offered that opportunity for you to be like, how about I just take a little bit of time and dive into yet another thing I'm not quite sure how to do, and then knock it out of the park. I think that that's so amazing. But I do want to ask, why did you decide to participate in the Codeathon? Mignon, would you mind sharing your experience of how you thought about why you signed up and why you participated?
No, I'm a neurologist and I specialize in neuroscience. And why I'm right now working in a Python lab and using a lot of coding. And I want to utilize my knowledge in neuroscience with coding, which I'm just in the studying phase with different languages. So I'm now year 2 master student. So I think it's a good time to practice what I learned from what I use right now in classes, in labs, into something like competition, which requires a lot of, members from other side of my current teams. So I learn collaboration, I learn from others, I learn how to join Python, and I learn how to collaborate and work out into a project with others. I think that's quite useful.
That's spectacular. Does anybody else have a question that they'd like to ask any member of the team or any of the teams themselves?
I mean, maybe just to throw it out there, another question. I know some people participated in our Codeathon last year as well, which was a much longer duration. Does anyone have any thoughts on whether they preferred longer versus shorter? Just like the challenge of trying to do cool stuff in a compressed time frame.
Go ahead, Christu.
Hi, yeah, I participated in the previous Codeathon. And as far as my experience goes, I would prefer the shorter format. I think even in our work process, it just expedited everything. We were supposed to deliver a product in 3 days. So we had to prioritize the things that would, basically have the package running rather than focus on things that would take a lot of time. So shorter format, I think, is the way to go.
Thank you.
Just to add to that, because just because of the experience that we had together in the same team, like the shorter format really did help making it so that we were not rushing off to do, because there was tons of ideas that we had, which we essentially said, if this is not working in the next 15 minutes, we are abandoning this. And that helped a lot in terms of making it so that we work on the ideas that are working, get it in a format that's usable to everyone, and not waste time on things that might not work in 3 days. And made it so that a project, like something that is actually deliverable and usable by others, is there, and not really something that is a jigsaw puzzle that others have to figure out.
Cool.
So Vindy, you're a first-year grad student, as I understand it, and was working under the time pressure of a Codeathon a new experience for you?
In a way it is, because I actually have participated before in hackathons, because I come from a computer science background. So, but those are like mostly 24 hours or like 9 hours ones. Like this is 3 days. So like we have to commit a bit more than 24-hour one. So what I thought was like I cover up some work beforehand, so I have more time in my hand for the Codeathon, and like limit a bit from the sleepy side. But because like we most of our members are like very keen on contributing, and like they all work really hard, so it was a smooth process, I would say. And I really enjoyed working with them, because it's not like one person is working and others are not working. So everyone works. So it's really nice.
That puts our longer codeathon into a different perspective, because last year's codeathon was nearly a month. And so going from a 9-hour codeathon, thinking that 3 days is a long codeathon, I shudder to think what you might think of the last year's codeathon. Well, I think we're at time, but I'd like to ask one more question, if I could, as long as no one else who is on the call would like to ask a question. But I guess what I'd like to know is if you could envision your tool being used by others, and if you have any advice for people just getting started working with the FAIR principles. So I'm not sure if anybody would like to tackle any component of that, but I think that's a good way to sort of wrap up our conversation today.
And so I can take the first part of that. How would I envision? I mean, one of the things I think Sparklet does quite nicely is that it allows basically users to add interactivity on like things on top of the flat map. So in some sense, it makes the flat map not just an end product of 50 different data sets into one, but also allows it to actually add on to those. So for example, one of the ideas, and this is, by the way, me reading from the Slack channel, because these are ideas that we had actually and discarded. One of the ideas that we had at the time was that, there are these kind of electrical recordings that are generated for different points in the nervous system, and what the Sparklet could do is allow you to actually take these electrical recordings and then put them together over the whole flat map, and then you can see, okay, that if you stimulate at a certain point, the other parts of the nervous system are reacting in a different way. So that kind of systems could be built on top of this. Other things that we were thinking about was that you essentially search for something and it allows you to get a PubMed list of resources, PubMed list, or like the whole Spark dataset list of the top 10 results, and then you can do analytics on that or just use the direct results themselves. That's the sort of things we were already thinking. Now, the last one that I would like to add, given that I'm basically working in a developmental biology institute, not a neuroscience institute, so I'm nowhere near close to neuroscience. Here, like one of the things that I really like that I saw in this particular project is that this project is not really tied to neuroscience as such, because if you have the flat maps, you can then use it for other things. So for example, one of my colleagues here is actually working on trying to map out, as a particular organism, say Tribolium, grows, how cells in it have different gene expressions and how they grow. Something like FlatMap would be FlatMap plus Sparklet could be really useful to actually visualize that process for biologists who are not really that much into machine learning, etc., because his code is really machine learning based. They could just import that and put their own data sets on top of it without having to know about all of this. Some of the cool ideas that we had at the time.
Well, I hope you participate in our next codeathon. Come back, bring your friends. And we do want to emphasize that these codeathons are really about bringing together people across multiple disciplines. This is for your neuroscientists and you biologists, as well as folks who have great interest in machine learning and unsupervised learning. Like, why not bring these folks together and utilize this set of data that is structured and organized in a way that you can take advantage of, right? So that's what we're hoping to demonstrate. We're hoping to encourage wet lab scientists to contribute their data to the SPARC portal so that it continues to grow and expand in the variety of data that it is housing, filling out that flat map. And then also giving that opportunity for more to be extracted from that raw data that's up there by letting all of you computer scientists go creative on it. So I'm really thrilled as a neuroscientist who does not code. I think you guys really did just a spectacular job, and I hope this is not the last time we get together and chat.
I mean, definitely, I will try to be there for the next one. And the information is already there in the institute, so expect more people.
All right, that sounds great. Does anybody have any last words before we call it a morning, evening, or afternoon, depending on where you are globally?
Maybe just one quick comment, just thanking, thank you, thank the SPARC team and the organizers of the Codeathon, because this was a really great opportunity. And I think we certainly learned a lot about the tools, but we also learned, I guess, the really great work that SPARC is doing. So we can sort of even potentially see how we can collaborate in the future. So this is awesome. Thanks very much.
Yeah, tell your friends, tell your mom, tell your grandmother about all the cool things that can happen with novel bioelectronic medicine, right? All right, thank you everyone for participating. And stay tuned, join our newsletter. We'll be announcing our next Codeathon via the newsletter. And thanks again for all your creativity and sweat and finger cramps from typing so quickly. Take care,
Thanks, everyone.
everybody.
Thanks, Sue.
Thank you.
Yeah,
Thank
thanks.
you.